Okay, so somatic cell fusion. So first we will see what is cell fusion, what is somatic cell fusion, and what is the application of somatic cell fusion. Any idea about this somatic cell fusion? Any basic idea? Reshma, Akshaya.
Sir, body cell ஒரு சில
என்னமா பாடி செல்லா
somaticனா normal cell, genomic cell இல்லனா
somaticனா normal
body
okay. Cell fusion. So what is cell fusion? Somatic விட்டுருங்க. Cell fusion என்ன? Okay, so as a name goes, somatic cell fusion is fusion நாவே ரெண்டையும் ஜாயின் பண்றது. அதான fusion. Fusion ஓட meaning ரெண்டையும் ஜாயின் பண்றது. Okay, so in cell fusion what we have somatic cell fusion, what we are going to do is we are going to combine two different species. Okay, so what we are going to do is we are going to break up the cell wall, and from the protoplast we are going to join or we are going to fuse two cells. Okay, for example, they were done in many somatic hybridization, like for example if you go for tomato and potato. Okay, so tomato and potato, they have joined these cells, and they have formed as tomato. Okay, tomato and potato, they have fused it, and they termed that as tomato. So this tomato will be having, so what is the use of this? Because we are having potato, we are having tomato, what is the use of cell fusion? We are going to we know about the characteristics of tomato and potato. Okay, so both the characteristics will be coming under this tomato. Okay, the characteristics of tomato and the characteristics of potato, both the characteristics will be in this tomato. So for that reason, and the other reason is for the disease resistant. Okay, for example, any diseases like for example potato leaf roll disease. Okay, so this somatic fusion will be resistant to this the tomato potato பண்றப்ப, for example, for reducing this potato leaf roll disease, this somatic fusion can be used. Okay, the main use is to study the disease resistant, to minimize the or to be disease free, and to increase the yield. அதுவும் ஒரு இது. Okay, நீங்க அந்த பனானா எல்லாம் பார்த்திருப்பீங்க. இல்ல பனானா பாத்தீங்கன்னா ஒரே கலர்ல இருக்கும். பனானாவோட கலர் தெரியுங்களா? என்ன பனானா கலர் என்ன? What is the color of banana?
Yellow.
அப்பா, வெரி குட். ஸோ எல்லோ நமக்கு எல்லோவா, பக்கா எல்லோவா இருக்காது ஃபுல்லாவே. அங்கங்க கொஞ்சம் கொஞ்சம் பிளாக்கிஷ் கலர் இருக்கும், அரல்ஸ் மோஸ்டா பிளாக்கா இருக்கும். எல்லோ தவிர பிளாக் இருக்கும். சில இது பாத்தீங்கன்னா இந்த டிசீஸ் ரெசிஸ்டன்ட் இதெல்லாம் எடுத்ததெல்லாம் பாத்தீங்கன்னா இந்த நம்ம ஃபியூஷன் பண்ணதெல்லாம் பாத்தீங்கன்னா ஒரே சைஸ்ல இருக்கும். சைஸும் பாத்தீங்கன்னா இர்ரெகுலர் சைஸா தான் இருக்கும். அப்ப பெர்ஃபெக்ட்டா அந்த பனானா சைஸ்ல வராது. அந்த நம்ம புக்ல எல்லாம் பாக்குறோம் இல்லையா, அந்த மாதிரி இருக்காது நார்மல் பனானா. ஓகே, இட் வில் பி லிட்டில் பிளாக்கிஷ் கலர் வி கேன் சி. அண்ட் இந்த மோரிஸ்னு சொல்லிட்டு ஒரு இது இருக்கு, நிறைய பனானா டைப்ஸ் இருக்கு இல்லையா. அந்த இதுல பாத்தீங்கன்னா அந்த பனானால பாத்தீங்கன்னா ஒரே கலர் தான் இருக்கும். மேலேருந்து கீழே வரைக்கும் பாத்தீங்கன்னா ஒரே கலர் தான் இருக்கும். சைஸும் ஒரே ஈவனா இருக்கும். அதை எடுத்து வச்சீங்கன்னா all the 10 or 12 bananas, if you are taking all that will be in a same size. Okay, so this is also one of the type of increasing the size, increasing the taste. Okay, and reducing the disease causing. So these are some of the uses of this somatic cell fusion. Okay, so in cell culture it is possible to produce hybrids. So this is called as hybrids. ஏன்னா நம்ம சில இதெல்லாம் பார்த்துப்போம். ஃபார் எக்ஸாம்பிள் இது கோவா பார்த்தீங்கன்னா, இப்போ பார்த்தீங்கன்னா இந்த சைஸ் பெரிய கோவாவோட சைஸ் தெரியும் நமக்கு. கோவானா கொய்யாப்பழம் சொல்கிறேன். கொய்யாப்பழம் பார்த்தீங்கன்னா இட் வில் பி இன் ஒரு ஒரு சைஸ் தெரியும், அந்த சைஸ் தான் இருக்கும். இப்போ வரதெல்லாம் பார்த்தீங்கன்னா பெருசா வருது இல்லையா? திஸ் இஸ் பிகாஸ் ஆஃப் திஸ் hybrid plant. Okay, hybrid variety plant எல்லாத்துலயுமே, not in vegetable and fruits also there are many hybrid fruits are there, so which will be like seed இல்லாம நம்ம பார்க்குறோம் இல்லையா? இப்போ கிரேப்ஸ்லேயே வந்து பார்த்தீங்கன்னா கிரேப்ஸ்ன்றதே வந்து it will be having 1 or 2 seeds, ஸோ அதுவும் சீட் இல்லாமல் வருது இல்லையா? ஸோ தீஸ் ஆர் ஆல் hybrid variety where they are removing the seeds. Okay, but that is not healthy when it is taken up for packing or when it is taken up for the tasteக்குலாம் வந்து it will be more tastier when compared to a normally grown the organic material. So in cell culture it is possible to produce hybrids of various cell types. Okay, so what is the process here is the cells will be fused in vitro. Okay, the in vitro it will be fused up, so it will produce either a binucleate heterokaryon or uninucleate hybrid cells. Okay, so these hybrid cells are employed to study the control of first one is gene expression and then differentiation, and then malignant cell growth, viral replication, and then gene mapping, gene studies, genetic modification studies you can do, and then production of antibodies. Okay, so these are some of the applications where we can use this somatic cell fusion. Okay, so hybrid cells were obtained for the first time in 1960 in France. Okay, from the mixed cultures of 2 different cell lines. So in 1960 is in France is the first hybrid cell which was developed. Okay, so the fusion of individual cells can be induced by Sendai virus. Okay, normally they will be using this this Sendai virus is an inactivated virus, not a virulent vaccine virus. Okay, so fusion of individual cells can be induced by Sendai virus and polyethylene glycol, and also by certain other chemicals they will be using for this fusion process. Okay, and in animal fusion of 2 different cells and production of hybrid cells have been successfully achieved. Okay, so these hybrid cells have significant applications, significant biotechnological applications in more areas. The first one is study of control of gene expression and differentiation. The second one is gene mapping. Third one is malignancy. Fourth is viral replication. Fifth one is antibody production through hybridoma technology. So these are the applications of these hybrid cells. Okay. Okay, so in bone cells, they were used for this kind of somatic cell fusion, where where the cells grown in culture are induced by some of the virus, such as normally they will be using this Sendai virus as an activator to fuse to form hybrids. Okay, so this virus induces 2 different cells, first to form heterokaryon. Okay, can you see this slide?
Yes, sir.
Okay. So this is a procedure where this somatic cell hybridization will be taken up. Okay, so cell X and cell Y. So different types of 2 cells can be taken up and then fusion. Okay, so after the fusion, we'll be getting that XY heterokaryon, and then the mitosis process, and then we'll be getting the 2 different hybrids. Okay, now you can take this in the note. எழுதுறீங்களா அக்ஷயா?
எஸ் சார்.
Cell fusion is a technique in which two different types of cells or cells from two different species are fused in vitro. This is done in the presence of chemical fusogens such as polyethylene glycol or by electric shock. Suppose these are two different types of cells, represented by X and Y. These cells are allowed to fuse by exposing them to chemical fusogen polyethylene glycol. The cytoplasms of these two cells combine and form a single large cell with two nuclei. This cell is known as heterokaryon. Hetero means different, and karyon means chromosomes. It is so called because this cell has two different sets of chromosomes. In the next step, this heterokaryon undergoes cell division, and the two nuclei also fuse. This resulting cell now contains a single large nucleus having two sets of chromosomes. This cell is known as somatic cell hybrid. Somatic cell hybrid is unstable. This is because of the presence of the extra set of chromosomes acquired during fusion. These extra chromosomes are lost randomly during subsequent cell divisions until the cell returns to the normal diploid state of one of paired chromosomes.
Okay. Have you seen that video?
Yes, sir.
Okay. So this is about somatic cell fusion. Okay. So the last topic is organ culture. Okay. And then, okay. So another two topics are there: valuable products from cell culture and organ culture. Okay. So that we'll do. So tomorrow is working, la? So tomorrow we are having class.
Sir,
Solpa.
sir, organ culture is also possible.
Yes, yes. Organ culture means they are not going to fuse
Okay,
both the
sir.
organs. They are taking up. They are going to take up the cells only, no? From the cells, we will be developing a new organ.
Okay, sir. Okay, yeah.
Yes. If you are taking any if you wanted to grow up an hand or leg or anything, from that the cells will be taken up, and then from that we will be fusing that, and we will be. So that is different. Okay, that is growing up of a particular organ is different from this.
Sir, it like
So this
heart
is
or eye.
see what is the difference between organ culture and this is organ culture means we are going to grow up the same organ. That's all. From the same cells we are going to grow up in a in vitro culture. But here somatic cell fusion is fusion of different species. Okay. For example, mice and then pig like that we are going to fuse up and we are going to see whether it is disease resistant and whether there is any problem occurs after the fusion like that we are going to see, and genetic modification also we are going to study in that. Okay, so how the genetic behavior between
Okay, sir.
these two different species like that. Okay, they have done many things in plants actually. In plant first they started with that tomato, I told, no, tomato and potato they have done. Okay, you can wait, I will show that we are. Whether anyone heard about this Pomato?
What, sir? What?
Pomato. Anyone heard about this Pomato?
No, sir.
See, this is a tomato. So this is a tomato plant where it has an see like that, like this it will be. So they have fused these tomato and potato, and they termed as tomato.
Yeah, sir. This has I have seen. Yes, sir. Tomato. Yeah. I have seen, sir.
Not tomato. Okay. Okay, you also you can browse and see.
Yes, sir. They used to mix that tomato and potato in same plant to grow the both of things
Yes, the same thing.
in agriculture.
Hmm. But there are many opposing parties. They are blocking this because it is from the natural thing. No? It's not a natural one. So we are going to make that. We are going to fuse that, and we are going to bring up it as a new plant or a new organ or new species. So that there are many opposing parties like BT brinjal. BT brinjal, you know.
Yeah, sir. I have seen this in flowers also. The rose and some other flower.
Yes. Okay, I will take the buttons. Answer attendance. Adit,
Yes, sir.
Akshaya,
Present, sir.
Anuja,
Present, sir.
Anuraja,
Present, sir.
Everton is there. Jennifer,
Present, sir.
Jennifer,
Present, sir.
Joel,
Present, sir.
Kavila Kumari,
Present, sir.
Kamachi,
Present, sir.
is it
Present, sir.
Nitish Kumar, absent. Priyadarshini,
Present, sir.
Raghavi,
Present, sir.
Reshma,
Yes, sir.
Sanjay absent. Sandhya, Sharat, Sandhya, absent. Sharat, Yachi,
Sir, absent.
absent. Taslima, absent. Ravina,
Present, sir.
Rishita, Sadia, Rishita, இருக்காங்களா இல்லையா? இல்ல absent.
Sir.
Shruti,
இல்ல, sir.
okay. Shruti, yes, present.
Present,
Sir.
Yes,
Sir, Sadia present.
Sai is present, yes. Surya Prathapan, absent. Absent. Bhavyashree,
Present, sir.
Present,
Is the
sir.
present.
okay.
Irkan.
Bhavyashree, present.
Present, sir.
Okay. Prashant, absent. Antika, absent. Priyadarshini,
Present, sir.
Sivanya,
Present, sir.
okay, ma, thank you.
Thank you, sir.
Thank you, sir.